The overall goal of this procedure is to obtain a pure population of human myoblasts from adult skeletal muscle. These cells are used for functional studies such as assessment of myogenic differentiation and measurement of store-operated calcium entry.
The main advantage of this technique is that it allows a rapid, simple, and reproducible isolation of human primary myoblasts. These myoblasts are then used ex vivo to study the calcium signaling during muscle regeneration.
This method can help answer key questions in the muscle field, such as identifying the ionic channels and the transcription factors involved during muscle differentiation.
For this method can provide insight into gene functions during human myogenic differentiation. It can also be applied to other studies, such as the potential for using myogenic stem cells to treat muscular dystrophies.
Transfer the muscle sample into a sterile 6 cm plate. Wash the sample with PBS twice. Remove any remaining adipose tissue and weigh the sample. Next, add trypsin and mince the muscle into pieces smaller than 2 mm in size. Once minced, use a 10 ml pipette to transfer the tissue into a 200 ml dissociation bottle containing a stir bar. Then continue collecting minced tissue in trypsin solution until the dissociation bottle is filled to 90 ml. Now, incubate the tissue in a water bath at 37 degrees Celsius for 60 minutes with gentle stirring. After an hour, add 10 ml of FCS to stop the reaction and homogenize the mixture.